Welcome everyone to this week edition of Extracellular Vesicle Club, the weekly event of International Society for Extracellular Vesicles. Today we have two amazing guests from Department of Cancer Biology and Molecular Medicine, Beckman Research Institute, City of Hope, Comprehensive Cancer Center, Dr. Tijana Talisman and Dr. Andras Saftics. They will present the recent paper in JEV, single extracellular vesicle nanoscopy. Welcome.
Thank you so much for this kind introduction, and thank you for having us. We've been following EV Club for a while, and we really learned quite a bit from these presentations. So it's really wonderful to be able to present today our work on single extracellular vesicle nanoscopy that was recently published in JEV. Andras, who is the first author on this work, very talented postdoc, will join me for this presentation. But I wanted to mention this was really a great lab teamwork. Sarah, Eugenia, Ima, Nan, and Katie contributed quite a bit to this work as well. And it was also an amazing collaboration with pancreatic cancer surgeon at City of Hope, Dr. Gog Singh, EV expert from MGH, Sumya Das, and his postdoc Mikhail, and another great EV expert from Tijana, Kendall Jensen. So just to briefly give you an overview about our technology, I wanted to give you the scale of biological systems. And you can see that there are different modalities for investigation of biological systems. Conventional microscopy is extremely powerful technique in cell biology because it allows optically based, minimally invasive observation of events on a really wide range of time scales. But the problem is a diffraction limit of about 200 nanometers. Super resolution microscopy techniques such as single molecule localization microscopy or SMLM can overcome this limit. And with these new advancements, they can go as low as about an angstrom. So it's really wonderful to see that. And microscopists love their acronyms. So instead of SMLM, you might have heard for Resi, Palm, Storm, or Paint. And I wanted to tell you that SMLM is truly a great a method for investigation of extracellular vesicles, first because it has this nanoscale precision. And nanoscale precision is important if we consider this lower limit of extracellular vesicles of about 30 nanometers. But it has additional really important property, and that is single molecule sensitivity. And if we consider cargo of extracellular vesicles, so if we want to go beyond the one vesicle and look what's inside of it, this is another very powerful aspect. SMLM is a fluorescence-based methodology. That means that we need fluorescent reporters that will label vesicles. And for that, we can use affinity reporters, we can use covalent reporters, or we can use fluorescent dyes that can intercalate the membrane of extracellular vesicles. But the way this technique works is really by separating imaging times. So effectively, we image only a subset of molecules in any one frame. And in front of you, you see this really pixelated image. And this is a fluoropore, and it still looks pixelated as you would expect it in regular microscopy. But there is information in these pixels, and that is number of photons. So we can fit this number of photons really nicely to find where is the most probable position of this fluoropore, and that's indicated here with this red cross. So what we really do in SMLM is we take these molecules, we image a subset of them, they turn on and off, they blink, so you have this molecular beautiful fireworks, and then they're bleached. And new molecules appear, they again turn on and off, and they bleach. And this process is repeated until we acquire all the molecules in this image. So you can see as I'm talking, this image of the extracellular vesicles is building up in time. And we wait until we acquire all the molecules to give you the story about this extracellular vesicle. In our experiments for our conditions, we acquire about 25,000 of frames. So obviously, I will not let you wait for this fireworks for here. And you can see how this vesicle started from a single dot, and you build this whole image. So our single extracellular vesicle nanoscopy, or 7SA, I mentioned we like our acronyms. It uses these wonderful properties of SMLM, so single molecule sensitivity and nanoscale precision, and adds the affinity isolation. So the size of extracellular vesicles really fits nicely within the resolution range of SMLM. But this technique is very adaptable and it has economical design. It really costs probably several dollars for a cover slip, depending on particular dyes or particular molecules that you want to isolate vesicles with. So we can start with a wide range of different EV sources. We can use biofluids, different biofluids. We can use cell culture. We can use excellent culture. And we can use either isolated or crude source of extracellular vesicles. And I think this is really important as we are considering what happens during the isolation process. And where this adaptability again comes in is the ability to affinity capture selected extracellular vesicles, so subpopulations that we are interested in. And then we can again fluorescently stain these vesicles with a wide range of reagents, so those that we are again interested in. And then we are going to image our extracellular vesicles. And then we are going to do processing and analyzing like we would do in regular SMLM. So just to give you a brief idea about the protocol, I wanted to mention that it's really robust. Data that is presented in this paper has been done by 5 operators in my lab. And we start simply with clean cover slip, and we attach affinity reagents to these cover slips. And in this case, on the picture, you will see we attached antibodies. To validate the method, we started with these recombinant EV reference material from N. Hendrix's lab that was really useful to us, and it's now commercially available. So these vesicles have abundant tetraspanin, CD9, CD63, and CD81 on their surfaces, but they also contain GFP, green fluorescence protein, so we can visualize these vesicles in diffraction limited microscopy as well. After we isolate or capture affinity these extracellular vesicles, we can stain them. And in this particular case, we used a combination of antibodies, again, tetraspanin, CD9, 63, and 89, T-span, that are labeled with Alexafluor 647, which is a red dye. This is really excellent for SMLM. And just to give you an idea how the images look like, this is a section of our region of interest, and you can clearly see the region where antibodies are deposited, and you can see these red clumps. Those are extracellular vesicles. And on the right side, you see pitch dark, and that is the spot where there was no antibody. So you can right away see on ROI how well this method works in terms of selectivity. And all through this presentation, I will show you raw images. And I think this is really important so you can kind of get an idea of the signal to noise of this particular technique. And the first thing we did is we counted the number of vesicles that we get with this technique. So we used several vials of these recombinant EV materials. We isolated them on the T-span antibodies. We stained them on the T-span antibodies, and we got really nice agreement between repeats. And we also tried a control isolation with regular IgG, and you can see that we can barely detect anything. So this was really exciting, but we wanted to make sure that we can validate our method with a complementary method. And in this case, we use TEM, and we wanted to check the sizes and shapes between the two methods. So on the top, you see TEM image, and on the bottom, you see combined SMLM and TURF image. So SMLM gives you this really nice, tight red spot where you see the vesicle, and GFP is imaged in TURF in green. So you can see that the spot is much bigger, it's pixelated, and that's because surface diffraction limited microscopy, but you can still get this wonderful overlap between two dyes. And in our experiments, we used Voronoi tessellation to analyze and detect vesicles, but we also applied another methodology, DB scan based using nanometric software, and we really got an excellent agreement between two methods. I will just show you the data that we obtained with Voronoi tessellation. As I mentioned, we wanted to compare the size and shape between TEM and SMLM, and you can really see excellent agreement. And because we have so many vesicles that we can analyze, you can see that we can get really nice heterogeneity of our populations as well. But one really big advantage of SMLM is the ability to count molecules. So what we did, we used SAMI method that allow us to calibrate our fluorescence reporter, and we calculated number of detected tetraspanin molecules on the CVs. And this is really an important additional aspect that we can get with this methodology that we can obviously not get with TEM. And now Andras will show you what you would see if you're in front of the microscope with a typical experiment, and give you an overview of this.
We recorded this video to show how a representative 7-assay surface looks like when we are scanning the surface using a low laser power. Here you can see a clear edge of the spot, while outside of the spot there is almost no EVs signal is detected, because just very few EVs could bound to non-specifically to the surface. The spot is very uniform, and crossing the spot, we can clearly see the other edge as well. Again, we have an excellent contrast in the signal between the inside and outside of the spot. To demonstrate the performance of our method, we used pooled human plasma samples that we thoroughly characterized according to the MESAV guidelines. We isolated EVs from crude plasma using size exclusion chromatography, and we collected and combined 5 fractions that had the highest EV concentration and highest purity. We further analyzed these combined fractions, and our transmission electron microscopy images showed that the EVs had intact morphology, and our dot plots showed that the EVs were expressing specific EV markers such as CD9, CD63, CD81, and syntonin, and we saw low abundancy of cytochrome C and ApoA1, which were co-founding proteins. We measured the bulk EV size distribution and EV concentration using nanoparticle tracking analysis, which provided an average concentration of 5 times 10 to the power of 9 particles per ml. After characterizing the SEC-isolated EVs using the standard methods, we applied these samples in our assay. We isolated these EVs on the surface, where we immobilized anti-CD9, anti-CD63, and anti-CD81 antibodies, and we stained the EVs using the same antibodies, which were stained with the Alexafluor, were labeled with the Alexafluor 647 dye. We collected raw images like this here, which demonstrates a great separation of the EVs and also a very low non-specific signal. We counted the number of EVs at different EV concentrations, and we found that the assay has a large range of linearity, covering at least a 64-fold dilution range. We also quantified the number of EVs in specific EV subpopulations. We isolated EVs on anti-CD9 antibody surfaces, as well as on anti-CD63 and anti-CD81 antibody surfaces, and we stained the EVs using the same antibodies labeled with the AF647 dye. We counted the number of EVs in the isolated EVs subpopulations, and in agreement with the dot plots, we found that the CD9 enriched EVs had the highest number of EVs per region of interest. We ran several controls, including experiments with non-specific antibodies, Triton X100 lysed EVs, and no antibodies on the surfaces. And each of these experiments provided a minimal signal. We also quantified the size and the detected number of tetraspanin molecules per EVs for the SEC isolated samples in the CD9, CD63, and CD81 enriched EV subpopulations. And also, we characterized the shape of the EVs, which was similar in the case of all of these subpopulations. In a two-color imaging, using a two-color imaging method, we could assess the distribution of the cargo proteins on EVs as well. In these experiments, we stained EVs with the red CF568 dye, with an orange CF568 dye, which was melamide conjugated. And in this way, it could stain the EV on the EV membrane proteins, which had available cysteine groups. Then we affinity captured these CF568 stained EVs on the surface, and additionally stained them with anti-tetraspanin antibodies with the red LXF647 dye. These are some representative images of the surfaces where we clustered the collected localizations in both the 640 nanometer and 561 nanometer channels. We identified co-localized EVs as clusters overlapping in both channels, and we determined the centroid of the clusters in the two channels. We plotted the centroid of the 640 nanometer channel detected clusters relative to the 561 nanometer channel clusters, which are here in this plot, the 0, coordinate. And we found that the centroid shift in the X and Y coordinate follows a normal distribution with an average centroid shift of 25 nanometer. Based on these results, the majority of the EVs were very well overlapping, and it is likely that those EVs which had a higher centroid shift, they had a heterogeneous tetraspanin distribution or clustering of these molecules on the EV membrane. We also demonstrated that the assay can robustly detect EVs directly from crude plasma, and it can quantify single EV characteristics of crude plasma EVs. Here you can see an image about the edge of an area where we affinity captured crude plasma EVs and stained them. Outside of this area, we detected only a very low non-specific signal. This is also represented in this zoomed in image, where you can see the clean surface and very compact EVs. This also shows that the Voronoi tessellation-based clustering could be performed with high reliability. We determined the localization precision of the assay using two independent methods, and we found that even in the case of crude plasma EVs, the localization precision was 78 nanometer on average. Using the crude plasma EVs, we ran the same type of experiments as in the case of the SEC isolated plasma samples. We found that the number of EVs of different EV concentrations had a linear range covering at least a 50-fold dilution range. This image, I want to highlight that it was collected on a surface where we applied only 0.8 microliter plasma, and the technique can go down as little as working with 0.1 microliter of crude plasma sample, and we just simply dilute the plasma. We also quantified the number of EVs in the CD9, CD63, and CD81 enriched EV subpopulations in the crude plasma samples as well. And we found that the ratio of the number of EVs per region of interest was well reproduced, as in the case of and observed in the SEC isolated samples. Our control surfaces, again, provided a very low number of EVs. Additionally, we quantified the size and detected number of tetraspanin molecules per EVs in the CD9, CD63, and CD81 enriched EV subpopulations for the crude plasma samples as well. As Tijana highlighted it before, we wanted to compare our technique with an established method. So using the crude plasma samples, we compared our assay with the ExoView technique. The ExoView technique uses an interferometric detection method for EV sizing and the combined fluorescent and interferometric detection for characterizing the CD9, CD63, and CD81 enriched EV subpopulations. In the case of the ExoView, the interferometric detection reported EVs in the size range of 50 to 200 nanometer. In the case of the 7 assay, we were looking at EVs in the size range of 30 to 400 nanometer, but this size range can be further extended towards the smaller and larger sizes as well, which may be important in the case of experiments where we want to look at these EVs which are smaller or even way larger. Also, the 7 allows for achieving molecular sensitivity, as we can detect the number of tetraspanin molecules per EVs per single EVs. We also wanted to compare the capturing efficiency of the two techniques. And to calculate this capturing efficiency, we normalized the number of EVs in the CD9, CD63, and CD81 enriched EV subpopulations. We had to do this normalization because the two methods are using different spot sizes and volume of plasma sample. We found that the capturing efficiency was in the similar range in case of the two techniques. As an important result, we compared our data acquired on the SEC enriched EVs and the crude plasma EVs, and we found that the SEC enrichment leads to a loss of about 4-fold of the EVs. This confirms that the isolation method affects the number of the detected EVs. To demonstrate that the assay works with other affinity agents as well, not just with antibodies, we immobilized lactate herin on the surface and affinity captured EVs, exploiting that the lactate herin is specific to phospholipids on the EV membrane. Compared to the lactate herin surfaces, the anti-tetraspanin antibodies isolated more EVs from crude plasma. This may happen due to the presence of particles with containing lipids in the plasma samples, and these particles can also occupy surface areas. Looking at the single EV characteristics, we found that the size of the EVs in the case of all isolation methods was similar. However, the number of detected tetraspanin molecules per EVs was significantly different comparing to the SEC enriched EVs to the crude plasma EVs. This might relate to the effect of some steric hydrogens, and this suggests that the SEC enrichment can reduce the EV corona. I'm letting Tijana coming back to introduce some great results with the PDAX experiments.
So now we showed you that we can definitely isolate and characterize vesicles that are enriched in these abundant molecule tetraspanins. But many of us are also interested in the vesicles that are really low abundant, those that are associated with a particular organ, tissue type, or those that are associated with a disease. So here we started to work on vesicles that are enriched in pancreatic ductal adenocarcinoma. And this is one of the deadliest cancers. Symptoms of PDACs are typically non-specific. Patients present late. Only 10 to 20% of patients are eligible for surgical resections. And that's the only potentially curative treatment options right now. So to date, unfortunately, early detection strategies are marginally effective. So what we wanted to do is to focus on patients that are eligible for surgical resections, and we wanted to look at vesicles that have markers that are relevant in that particular case. So we focus on two different markers. First one is IGF-1R, and this particular receptor has higher expression that is associated with worse outcome in patients in resected PDAC. The second one is CD9. Interestingly, CD9 is highly expressed in PDAC microenvironment, and particularly in stroma, and its abundance is associated with poor prognosis in PDAC patients. So I first wanted to give you another raw set of images to show you how these vesicles look like. So you have a healthy subject, and you have patients that have PDAC P1 to P4. And on the top are CD9 enriched vesicles, and on the bottom are IGF-1R, insulin growth factor 1 receptor enriched vesicles. What's really obvious right away is that even by a naked eye, those IGF-1R enriched vesicles are larger, right? They are much less abundant. Now, these images don't show that, and that's because we use very different dilutions for these experiments. But when we back calculate, only 0.5% on average of EVs are IGF-1R enriched. And this is really wonderful to show the sensitivity of technique to capture these rare population of EVs. Many of us are interested for all sorts of experiments, including biomarker discovery or looking at therapeutics and even in cell biology. Now, we wanted to analyze these vesicles, and we did this using our methodology. And I wanted to first show you CD9 enriched vesicles. And we compared ExaView and 7 again, because ExaView right away gave us a hint, right? There was average diameter of about 60 nanometer for healthy subjects, where these PDAC patients had lower values, right? But then we applied 7, and we really saw nice differences. We saw that healthy subject had average diameter of 91 nanometers, whereas it was significantly lower in those PDAC patients. But where we really were interested is looking at those IGF-1R enriched vesicles that are very rare. And in this particular image, you see a bunch of dots, and each of these dots represents a vesicle and gives us two values. It gives us on x-axis diameter, and on y-axis, it gives us number of detected tetraspanin molecules per EV. And on the bottom and on the side, you will see the box plots to give you the heterogeneity of these populations as well. So for the healthy subjects, so what we got is about 17 detected tetraspanin molecules, and we got size of about 86 nanometers. But when we looked at PDAC patients, we really saw a sharp change. We noticed that the size of these vesicles is significantly increased, and we noticed that number of tetraspanin molecules is also increased. And lately, there was a lot of papers that are mentioning the shape of vesicles can be perturbed in cancer. So we wanted to see how the circularity or roundness of vesicles compares between healthy subjects and pancreatic cancer patients. And what we indeed observed is that there is a significant difference in circularity between the groups. So pancreatic cancer patients had more circular vesicles. And just to show the utility of this methodology for the biomarker discovery, we wanted to select those vesicles that have higher diameter and higher number of tetraspanin molecules, and we wanted to quantify them. So these are these vesicles that are encapsulated in the polygon with a dashed line, and then we count these vesicles, and indeed we see pretty nice differences between healthy subjects and those for pancreatic cancer patients. And of course, this is just the start. We wanted to show you that this methodology is indeed very useful for these type of experiments. And I hope that we showed you today that 7 can really comprehensively characterize individual EVs. It gives you many parameters, and you can compare all sorts of different properties. You can look at number of extracellular vesicles that carry a particular marker that is of your interest. You can look at the size of these vesicles, at the shape of this vesicle. You can look at molecular cargo content, and if you have a good probe, you can analyze anything that you want to. So there are some images here. You can look at lipids, you can look at microRNA, you can look at luminal proteins, right? You can look at heterogeneity of these EVs because you have really nice set of populations, and you can look at how they're distributed, and you can also look at distribution of markers. And Andras has shown you that on example of how tetraspanins are distributed on the surface of EVs. And of course, this is a really big effort from my whole group that put a lot of hours in this, because we wanted a method that can be good for many other groups and people can use in the future. So Andras led this work, as I mentioned, but many people in my lab contributed. Sarah, Nan, Ima, and Katie and Eugenia are our alumni. And of course, this is a big collaborative effort. I'm immensely grateful for our funding agencies that helped us do this work. We can't do this without them. And I'm extremely excited to be part of ERCC2 NCATS consortium, because investigators in these consortiums also helped shape this work. So thank you for your attention. We will be happy to answer any questions you may have.
Well, thank you so much, Tijana and Andras. We do have some questions that have come in the chat box. And so if you have not placed your question in the chat box, please go ahead and do so. I am currently in the process of allowing people to unmute themselves and start their video. So when I call on you, please step up and ask your question. So I just want to note, we did have a few answers to questions in the chat box already. And thanks very much to members of the Talisman lab who helped with that. But we will go through those questions anyway, just because the people on YouTube are not going to be able to see the chat box. So let us start out then. I have a question from Randy Stout. Please go ahead.
Hi, that was really great to see all of the characterization and comparison work that you've done here. I really, it's so exciting to see that because of the power of this technique. I have two quick questions. One is when you refer to the 7 technique with this acronym, is this all single molecule localization with capture, or is that specific to using tetraspanin antibodies to adhere the EVs to the cover slip?
So you do not need to use tetraspanin. So Andras has shown you that you can use lactoferrin. You can use any affinity reagent. We used IGF-1R to look at PDAC patients, and subsequently we have done many different isolations. So it works with any affinity reagent that is capable of capturing vesicles. And again, you can stain any which way you like. So it's a mix and match, and that's really the power of techniques. So thank you for your question. That's really exactly what we were trying to do. It can work with anything that works for your particular research.
And I have a very quick follow-up, hopefully a very quick follow-up from on that. So it was amazing to see this with the comparison between size exclusion and crude serum. Do you think that because it's actually in this case able to, if you use the ceramide dyes or something, see how much cellular debris you're getting versus true EVs when you co-stain with a tetraspanin? And in a lot of cases, actual crude serum is going to be better than using any kind of other further purification step, because you'll see things that you would otherwise lose or miss, and these low abundance EVs, for example, on top of getting size and shape information and things like that. So do you think that there's some kind of a discussion that should go on about, if you're using this technique, single molecule localization microscopy, how it changes the required recommendations with regard to purification or characterization of EVs? Because again, another example is that you have a larger dynamic range on the size than using the interferometric method, and you get better in some parameters, better information than using NTA. So what are your thoughts on the comparison of going forward, now that you've done all this characterization, how we can use this to characterize EVs? I guess it was a long question, sorry.
Yeah, that it's a great question, right? I don't think there is a right or wrong. I think you use what works for your research. I prefer looking at EVs directly from crude plasma, but I also understand that that comes with certain caveats. And caveats is that, there are situations where your molecules of interest could be included with corona proteins or sterically hindered, or there's a lot of other molecules around it, so it's harder to access, right? So sometimes you will have to. There are situations where you don't, right? So I think there is no recommendation, except I prefer, when possible, to look at crude plasma, but we typically compare things and make sure that things are making sense, and also make sure that good affinity reagents are used, because you're as good as your reagent.
Thanks for the questions. Christina Catoni, you have a couple of questions. So let me just ask these questions then. Which kind of affinity-based EV kit did you use? And I believe there's an answer here that there wasn't a specific kit, right? You're just using.
You don't use kits.
Yeah.
That's kind of the thing. You use what works for your research. We actually very clearly identified antibodies that we used. Use lactoferrin, we used the IGF-1R antibody. So use antibody that you validated, that's good, that works for you. That's the trick. You don't use kits. That's why this is really an affordable approach.
Yeah. So you can just put whatever you want on there as long as the chemistry is appropriate.
That's exactly right. And obviously it's selective agents. So again, you're as good as your reagent here for isolation. But you can mix and match, and that's wonderful.
Yeah, and so Christina's second question, and yes, Christina does not have the microphone, is, are you using Elements software? What are you using to analyze?
So that's a great question. So obviously, I have an Icon instrument measuring, but any SMLM instrument works. You have to make sure that you optimize your imaging to get excellent precision, to characterize your biophysical properties of dyes, which we very carefully do in the lab. And then you can do all these type of assays or analysis. So what we typically do, we process the data using this Elements, and then further on, we use MATLAB to use either Voronoi tessellation processing, or we use nanometric software that has pre-built DB scan based algorithms to also analyze them. You can use what works for you. Again, that's why we wanted to compare two different types of analysis method to show you that it's really comparable. So what works for your research should work here. So you kind of have to do processing and then analysis separately, because you need to get localization with the first software and, obviously use the instrument that you have in your disposal.
Great. All right. Minglin Liu, please go ahead with your question.
Great work, great talk. Have you ever tried to use Anderson 5 to capture the UVs, then scan it with scope?
An XN5?
Yes.
We tried once, we got fewer EVs, very few EVs at that point, but we are not stopping, right? Because it was just a very quick try once. But effectively, as long as the affinity reagent captures EVs, you can do these experiments.
Yes, because in early time, that time, of course, like 10, 20 years ago, the study mainly based on the, Anderson 5 to count as a marker of the EVs. At that time, the literature, based on the flow cytometry analyses, people say 2/3 or 3/4 of the EVs has a phosphatidylserine exposed on their surface. So if myself, we did not do this, but I heard people use that to as either negative or positive selection of the EVs, use coated the kit with plate with Anderson 5. I think that's worthy to try.
Yeah, definitely. And like that heron works in a similar fashion, right? So that's basically what we used as another way to show it's not only antibodies, and you can select based on the phospholipids on the extracellular vesicles. So that's a great point. Absolutely something that we can do.
Okay, thank you.
Thanks, Minglin. And I realize now that I also skipped over some people, so I've been hopping around in the chat box. Melanie Schurtz. Okay, Melanie might not be there.
Yeah.
Oh, there you are. Hi.
Okay. So it took a bit. Thank you for this nice talk. I think my question was already answered, because I was a bit confused with the image that you had. Okay, the binding of the antibody, and then really a sharp line with non-binding. And I thought it's about your treatment of the cover slip. And I thought, why is it not binding to when you don't have treatment? Because we see that there are a lot of EVs just binding to glass surface or other surfaces. But I think it was answered that they still bind.
No, actually, we block that area. That's why you see pitch dark. So Andras showed you this nice movie where there was a pitch dark and then there was an antibody spot. The reason why things do not bind is because we first clean the cover slip and then we really make it as a non-fouling surface. So it shouldn't isolate vesicles, right? So we cap it so it should not bind. So that's why it's working so well. So it is non-fouling surface around the antibody, and the antibody is there to capture vesicles. So basically very, very low binding that is non-specific, because the way we prepare these cover slips, it's really excellent. That's why I wanted to show you the movie. But the way we affinity isolate on unlabeled antibodies, that was one of other point that you raised, and then we stained with a labeled, fluorescently labeled antibody. So your surface is not gonna have on its own any fluorescence. It's only once you isolate vesicles and then label them. I hope that answers your question.
And how is it that you are blocking? What's that?
Thanks. I wasn't aware. Yeah.
So we use the polymer from Lucident that has NHS esters, and we just block those NHS ester spots with a blocking solution, and then they're nice non-fouling polymers similar to PEG, right? PEG should work as well. So you should not isolate any vesicles on the PEG-coated cover slip. Similarly, these polymers work really well once they're capped. They are not going to have any vesicles not specifically attached to them. Does that make sense?
That's great.
Yeah, that's cool that it works so well.
Really... Yeah, that's what we wanted to show the picture. Yeah.
Well, thank you.
All right, let's move next to Surya Aryal.
Hello, very nice talk. And I think this is a very powerful method for EV characterization. And I saw you were using T-span based capture, and you also talked about a phospholipid based method, lactidihydrin. And somebody had a question about this phosphatidylserine, and like looks like it can be used in different methods. So I was curious, like if we rely on T-span, is that like would we miss a lot of EVs which are not T-span, not expressing T-span? Based on your experience, do you have any idea like what proportion of EVs, let's say in plasma sample, would have T-span expression which we can use to capture them?
So that is a very important question, something we think often about. We do detect T-span positive EVs in this particular case, but you can mix and match. But what do you call an EV, right?
Yeah.
If it has, the membranous portion, you can isolate particulates or sheds from the membrane that are not vesicles, right? The same way. We tried several dyes. They're working, but not as well as we would like to select the vesicles. So it's always, what you use to define a vesicle. So you're absolutely right. We will miss those that are not enriched in tetraspanins. Sometimes we apply additional antibody. Let's say that I'm interested in a cancer EV that has an expression of something that is very abundant. I can use an additional antibody, because there were papers that are saying that these tetraspanins could be getting less abundant in cancer, right? So you can use other labels. If you have a good idea what you consider an EV, you can use a dye that works well. Remember, in SMLM, molecules need to blink, turn on and off, so not all dyes are going to work as well. So you have certain limitations there for dyes that are intercalating and that you would define as a vesicle. So it's a great question, something I think about. That's why we always say T-span enriched, or whatever you use for enrichment. I think that field can really work on that to define better dyes that are universal.
Yeah, very nice. Thank you very much.
Thanks for the question. Danilo Mladenovic, you asked about reference materials, and we heard about that fluorescent kind of virus-like particle. Does that answer your question, or do you want to follow up?
Sorry, maybe I missed this information, but yeah, actually, I was actually hoping to hear how this cargo molecule quantification was done. So you use some reference material for this?
So the reference material we use are basically vesicles that Anne Hendricks reported in her Nature Communications paper, but they're now commercially available, I think, through Sigma, right?
Yes.
Yeah, and you can just purchase them, and they have, lots of GFP molecules inside. But in terms of how we count cargo, we actually make sure that we properly characterize our fluorescent reporters. So we would put fluorescent reporters on the cover slip and look at their blinking behavior, because we want to make sure that we know what is the number of localization per fluorescent probe, what is the maximum dark time, right? So we really care about these biophysical properties to count properly. So you actually have to do this extra step to count.
So according to the single antibodies that you're looking at on the cover slip, you're basically using that to convert the fluorescence intensity.
That's exactly right. And that's very clearly described in the paper. And I gave the reference to our previous JAX paper where we show that works really nicely. So as long as you have isolated spots with antibodies, so everyone has to do this, right, for their microscope, their illumination conditions and so on. Because if you change your degree of labeling of antibody, that number is going to change. So we really tightly control that. So you actually also have to think about maximum dark time. You want to make sure that your dye is nicely transferred back to dark state, and that you're imaging longer than the maximum dark state typically to get all the information that you need.
All right, great, thank you.
Next question from Syed Iman.
Yeah, hello.
Hi.
Thank you very much. Hi. Thank you very much for the talk. It was very interesting. And yeah, it seems that this antibiotic capturing immobilization of EVs on the surface is going very promising method for detection of EVs. I've just had a question that did you have any TEM comparison between the crude plasma sample and SEC samples, the ones that is separated using the size exclusion chromatography columns?
So we couldn't obviously get EVs directly from crude samples, but we actually did TEMs on these purified EVs, and Andras has showed that, so they indeed have the intact morphology. Now, if you're using those, you need to make sure what you define as an EV, and certainly the size ranges were very comparable if you look at those vesicles that we report and many other images that we have, but you have to purify them by size, right? Or any other way, because you're going to have too much debris to image them properly. So you can't really do that from crude plasma. And that's really the advantage of this, is you really don't care what is your source.
Okay, yeah, because as I understood that, what is the concern that is raising these days is that in the biological real samples we are facing also with the lipoproteins LDL, HDL, that they also carry tetraspanins, and it's a possibility that we are detecting them instead of EVs. So yeah, the TEM
typically don't have as many tetraspanin molecules, to best of my knowledge, right? And because we get the molecular count as well, I think that helps.
Makes sense, yeah.
Yeah, I would just add it too that when we are analyzing our images, then we see the single EVs on the images. So this is an advantage that you can decide like how an EV should look like, and based on this you can analyze and identify EVs.
Okay, great. Thank you.
Thank you. Kevin Clayton, I love your question because we struggle with this in the lab too. Go ahead.
Hi, can you hear me?
Yes.
Yeah. Well, I guess I'm happy to hear that we're not the only ones. I guess, yeah, so the question is, do you have any advice on kind of what fixation and permeabilization conditions should be to detect cargo proteins, such that, say, you're capturing and you, without permeabilization, there are markers that are not revealed, but after permeabilization, you can't see them, because we've tried many different conditions and different kind of fixation conditions, and using different surfactants, say like Triton and Tween, at different concentrations, and we haven't really landed on anything kind of the concrete yet. And it's to the point where I'm kind of worried that, it's kind of easy when you're working with cells, right? Because cells are enormous in comparison to antibodies, but antibodies are, depending on the size of the EV that you're looking at, an antibody is literally a tenth the length of your EV. And it makes me almost believe that it's going to be very difficult to permeabilize an EV such that you can finagle your antibodies in there efficiently. And so, well, anyway, do you have any kind of advice or
so I can give you a good advice on the fixation method, because we really have that tested and done properly and happily, because we can compare stuff and you're very happy. And we use 4% paraformaldehyde with 0.2% glutaraldehyde, and we use obviously the best quality. You need to quench that if you're doing SMLM to make sure there is no background, and we do it at room temperature for 30 minutes. So that is a really nice way. And there are several papers, like early SMLM papers, like the work by Kusumi. When I was in Jennifer Lippico-Schwartz lab, we also looked at GPI and used the same fixation method to show that things are really not moving with that. So we know that this is efficient for fixation. Now, the second question is a really hard one, and we are struggling with that too. So we published a paper where Katie Lennon was the first author when we looked at troponin T, and we permealized vesicles in solution with twin-20. That worked. It's a really long protocol. We are still trying to make this work better, and it's tricky. It's a really tricky thing, and I agree with you. When we used microRNA, we used beacons that had cell-penetrating peptides, so that worked nicely. Getting antibodies, I think, is going to be challenging. As I said, we don't have the answer for you. We are still working on it. I can tell you that this protocol that we published was 0.05% V20 in solution, and that works way better than saponin that gave us a lot of debris. Much more twin would start permealizing, breaking the EV, because we can actually look at the size, right? So if you see that the size is going down and number of particles that you detect is going up, it's indication that EV is being broken. So that at least you have this kind of control built in. But yes, permealizing on the surface, we still haven't done properly. I struggle with that. We use tricks like CPP peptides for beacons, yeah.
Okay, thank you.
Okay, we have a few more questions. Let's go back to Christina first, then Randy, and then Phil. So Christina asks if you have tried an antibody of cocktails, so for the detection. Have you ever mixed a bunch together, and if so, how many could you put together?
Yes, so it's a great question, because we do worry about the affinity differences between antibodies, steric hindrances, and all sorts of things that are important in this particular case. And we did a maximum of I would say I know a case of 6. But we do it in 2 colors, like free plus free. That's probably the maximum we have done in the lab, and Ima is working on that. But the point is that there could be a competition, right? There could be a competition, but what I'm saying, I'm detecting tetraspanins. So I'm using a certain amount of CD9, 63, and 81 typically. Would that change? Could I have the individual one possibly giving me different data? But I want to encapsulate the whole vesicle, right? So that's why I'm doing it this way, because I want to make sure that I get the full size, and that's why I'm using all 3 of them at the same time. So you saw that there are some EVs that are patchy, so I'm avoiding this patchiness by using 3 different tetraspanin molecules. And there could be some competition. So you have to think about it, and you have to match it for your question, right? So as long as your question is making sense, I think you're okay. But is there steric hindrance, and is there competition? Yes. You know, if you have a very high affinity antibody, if you add another one, you're going to have a problem.
So it might be necessary to really explore what the implications of that
that's
mixing are.
exactly right. I think it's a specific for a particular set of conditions that you're using. Yeah.
Tijana, Andras, are you able to stick around for another 5 minutes?
Yes.
We do have we still have a few questions, so we'll go to Randy again. So Randy, you have a few more questions here. Please go ahead.
Hi. Yeah. So have you looked at the effect on size or circularity and just the shape of these vesicles on whether they've been frozen down, or they're fresh, or if they're lyophilized?
So that's what we want to do. This is a very important question in the field. I think that we need to have better handle of how we purify, how we freeze thaw. Ben in my lab is planning to do that, and absolutely something that needs to be done. And you know, there was again a recent paper in JEV, and it was featured here at the EV Club, that uses different buffers, right? So all of these things matter.
And you mentioned that you've been exploring different membrane intercalating dyes. What's that's been a little disappointing for us so far. We've tried a few different ones, and what's your take? Is there a best one? Are you still looking for a better?
I probably tested over 10, maybe 20. So basically anything that comes out, there are some that are okay, but nothing so far that I tested and I can say, hey, that works great. I use WGA, right? But WGA will stain more than an EV. And that goes back to the question, what is an EV, right? How do you define, or EVP, right? I care about these things. So that's why I try to use the language that really showcases what I'm doing. I didn't find the best dye.
We've tried WGA, and of course with the tetraspanin co-marker, and the correspondence was nowhere near the level that it was okay, but it wasn't something that we could reliably just use WGA at all. And that's what we were finding, at least on our side. But
effectively, nothing else worked as well as tetraspanin mixture for us. Let's put it that way.
Interesting. All right. Phil, you have a couple of questions here.
I'm sorry I came late, and this may not, I had the wrong link in the.
Oh, glad you could make it anyway. Thank you.
Yeah, my questions concern RNAs. For individual EVs, what percent have any RNA, and what percent have microRNA? And do individual cells make homogeneous EVs?
That's it's a very hard question, but it's a question that I care deeply about. If you want to use RNA dyes, they are typically not blinking. So it's really hard to use this methodology to ask this question, no matter how important I find it, right? We used microRNAs, and I can tell you that beacons that we use depend on a particular cell population. Sometimes if I have a specific beacon that's very abundant, I can get, 15% or so. But typically we have low values for the things that we have tried. Anecdotally, I see RNA in not as many as I would expect to see. But again, that is dependent on a dye or a particular probe that you use to detect an RNA. And in terms of homogeneity, I think they're extremely heterogeneous, right? So we take vesicles from a cell line, or not even going to actual biofluid, and the size ranges are enormous, right? The tetraspanin content ranges are enormous. So I think they're extremely heterogeneous. I think that not as many as I would expect have RNA, but again, I could not be using the optimal probe. And that's a great question. I really deeply care about that.
Well, it's a general knowledge that there's very little RNA, but they can transfer RNA to other cells. So that's a challenge for our thinking.
Yeah.
All right, well, we will end on that challenging note. Definitely much more research to do, and thank you so much, Tijana and Andras, for bringing these tools to us and sharing with us what you can do with your 7 setup. So thanks everybody too for joining and for the great questions and discussion. And we hope to see you again soon at a future EV Club. Thank you all, and take care.
Thank you so much. Bye.